Okay, so this audio lecture will be divided into two parts. The first part will look at lymphocyte receptor genes, which is chapter 6, and then the second audio lecture will look at MHCs, which is in chapter 7. And just to refresh your memory for some of the information from chapter 3, where we talked about the B-cell receptor, with the B-cell receptor, it has a transmembrane when it's a surface receptor on the B-cell. But we can also have it where it's cytosolic as it's being expressed, and then it also can get secreted. So we'll talk about how this process happens, mRNA splicing, that we go from having something that is membrane bound to a secreted form. And you'll remember that when we look at the B-cell receptor, or when it's secreted and we call it an antibody, there are different regions on it, and that we have these variable regions which the antigen would bind to. So those regions are unique to that B-cell, so that it would be able to recognize a specific antigen. So this is what changes between each B-cell, between each antibody, but the constant regions stays the same for the class of antibody. So the heavy chain and light chain that is utilized for a specific isotype or class of antibody would be constant, hence the term. Okay, so what we'll end up seeing in this lecture, in this chapter, is that that variable region has pieces of different gene segments, and that's how you get that specificity, that binding to a specific antigen. So if you look on the right upper figure, if you look at that variable region, you'll notice that there's different colors in there. So there's portions that are from the VH region, there's portions that are from the J region, and then for the heavy chain, there's also portions from the D region, gene segment region. Okay, and then for the T-cell receptor, that we have different domains within our subunits, so that the T-cell receptor is made up of 2 subunits, and then within that, they each have a constant region and a variable region. And again, the variable region is what's going to code for what peptides, what epitopes, antigens they can bind to. And that there's 2 T-cell types. There is alpha and beta, or there is gamma delta. And that gives us a lot of additional diversity in what antigens can be bound. So then looking at the chapter 6 material, there's this big question of how can we recognize so many different antigens in unique portions of those antigens with the B-cell receptor, but also with the T-cell receptor. Beginning of this lecture, we'll focus more on the B-cell. If we look at the B-cell receptor, there's over 10 million different combinations that you can get, and it's actually much higher than that. It's just doing some basic looks at how these genes can come together. Over 10 million different possibilities of what that variable region could be, and again, that gives us the ability to recognize all of these different portions of proteins, right? Or not just proteins, but DNA, whatever's going to act as the antigen. And so the question becomes, well, how can they do that? And then there's experiments that were done in the 70s that kind of elucidated how the B cells can actually accomplish this. And the way that they do this is by recombination of gene segments to end up creating the different possible antibodies and B-cell receptors. So there are different gene segment regions. So we have the variable, which is abbreviated V, diversity, which is abbreviated D, joining, which is abbreviated J, and then constant, which is abbreviated C. Okay. The D, the diversity region, is only found in the heavy chain. So we have genes that are for the light chain, and we have genes that are for the heavy chain. And so what you're seeing in this figure, the figure that's to the left, is your light chain, where we have different variable regions to kind of pick from, and then we have different J regions to pick from, but then we have a constant region that is specific for that light chain, okay? And then on the right side, we have our figure for the heavy chain. So again, we have a number of variable segments to select from. We have D segments to select from, J segments to select from, and then we have a constant that is the same. So we have one constant for that heavy chain, okay? So that when we end up undergoing, when the B cell ends up undergoing recombination, they're going to pull a J, a V, a J, and then that constant region stays, right, for the light chain. For the heavy chain, we're going to pull a V, a D, a J, and then we're going to keep that constant, okay? And then if you think about all the different kind of possibilities that you could get from, if you use your first variable segment and your first J, well, what if we use our second J with the first? And so you get all the different combinations, and that's how you very quickly can get up to that, not quite the, 100 million, 10 million, because there's other things going on that we'll talk about, but that gives you a lot of variability, a lot of diversity. So when we look at the proteins that will end up making up our immunoglobulin and our antibody, we are going to have 2 identical heavy chains and 2 identical light chains, okay? The light chain can either be kappa or lambda, okay? So one of the things is that these genes aren't kept together. They're actually found on different chromosomes. So for our light chain, our kappa genes for humans are on chromosome 2. For mice, it's chromosome 6. For lambda, the genes for human are on chromosome 22. And for mice, it's on chromosome 16. And then for the heavy chains, that is on chromosome 14. And for mouse, chromosome 12. And so again, the kind of take home of this is that it's not that all these genes are found right in the same location. They're found spread across the genome. And so that we are going to one location or another location, right? And we have to have heavy chain and light chain in order to make that. So regardless, we're going to 2 different chromosomes in order to make a single B cell receptor. So for the kappa light chain, we have the V, J, and C segments. So again, with light chain, there's no D segments. And with each of these segments, except for C, there are multiple genes that are found, right? And so for mice, there are 18 gene families for our V kappa. And then for the joining kappa, there's going to be 4. So humans have a similar arrangement, but different numbers of V and J kappa genes. So with lambda light chain, the gene pairing is a little different, so that there's different C segments and they are paired with J segments. So those go together. So for the variable region there, for humans, there are 33 variable genes that are used, and then there's 7 of these J-C pairs, but only 4 of them are actually functional. With humans, about 40% of light chains are the lambda, meaning 60% would be the kappa. Inversely, with mice, only about 5% of most IgGs have lambda light chains, and that's because they don't have as many variable regions, so they only have 3 functional variable regions. So there's a shift to like about 95% of most IgGs, the light chain being the kappa form, because there's more variation among it. And then looking at the heavy chain genes, again, we have V, D, J, and C segments. So there is that D segment that's present in the heavy chain that is not present in either of the light chains. For humans, there's at least 45 functional variable segments. Mice have 101 variable segments. And then just downstream of the variable segments, the V segments, we have the D segments. There are 14 D segments in mice and 23 in humans. And then for the J segment, there are 6 in human and 4 in mice. And then when we look at the constant region, there are 8 constant regions. And that's because we have the different isotypes of antibodies. So again, if you had like an IgG, one isotype, it would have a certain constant region, where IgE has a different constant region that it utilizes. So looking at our different chains, this is showing you the kappa chain. Again, you have your different variable regions, your joining, and then your constant. And if I were to put this figure onto the exam, and I said, is this a light chain or a heavy chain, that top one, the easiest way to tell is that there's no D, right? So in panel, in the bottom one, so it's marked heavy chain DNA, you can see how you have those D segments, okay? And so that lets you know that that's the heavy chain. You'll also notice that the additional constant regions, and with those additional constant region, that's so that you can have the different isotypes, different classes of antibodies. The way to tell kappa and lambda apart is, again, lambda is going to have those joining regions, segments that are right next to the constant. So you can see how the spacing, if you will, is a little different. The setup of the gene is different, that we have these variable regions, but then there's joining regions right next to constant regions. And so the variable region of the gene segment allows for diversity, and so there's that recombination that occurs with the various V region gene segments, and that's what's going to give us the diversity of that of that antigen binding site of that variable region. So you can see in this figure, which is not from your textbook, but it nicely demonstrates how that variable region ends up being made up by these different gene segments. And that the variable region of both the light chain and the heavy chain contribute greatly to what ends up making up the variable region of the immunoglobulin. And again, this is something that when I can annotate, you'll be able to kind of understand a little bit better. You do see that the J and D gene segments do contribute to it also, but again, there's lots of variability in the V segments that can be selected for. So one of the things is, I don't expect you to memorize how many segments there are for each of these different regions on the different genes. So, I'm not going to say, oh, which one has 7? So that's not what I'm looking for here. What I want you to do is kind of keep in mind some of the different characteristics of these different. So what makes kappa different than lambda? And then the big piece here is when we look at these different segments, the more segments we have, that ends up the greater the number of combinations that we can have, right? So in this table, we're looking at our heavy chain segments, our kappa chain segments, and our lambda chain segments. And if we keep in mind the different segments, constant's going to kind of be selected for, or for lambda, it's going to be linked to J. But it's really that variable region, you can see how in all of those chains, the greatest number is in the variable, right? And so each one of these variables can pair with, for the heavy chain, with the D and the J, right? And so for our light chains, each one of these variables could pair with the J's, right? And so we get all these different possible combinations, and so you can see how we could do the math probabilities of pairing and pairing heavy chains with each of these light chains, and that's going to further increase the number of possibilities that we could have. And that in itself gives us a lot of variation that we could have. Okay. Again, it doesn't quite get us to that 10 million different 10 million different possibilities, but it definitely is getting us closer to that because of all the different possibilities of which combinations we get. Now, what's the mechanism? How does this actually work? How does VDJ recombination occur on the heavy chain and VJ recombination occur for the light chain? The way in which these segments are kind of identified is by recombinant signal sequences, RSSs, and they flank each of these gene segments. And so again, it gives our enzymes a marker, a sequence to look for, to bind to, so that they can identify what's the good piece, what part do we want to keep, so that we're not splicing out in the middle of a gene segment. So that each one of these is conserved in either a nanomer or half-mer sequence, and then in between those lies these spacer sequences. Those spacer sequences are either 12 to 23 base pair spacer sequences. And then, so that's what ends up being our recombinant signaling sequence. So looking at this on one of our genes, we can see how we get these 12 base pair spacers or 23 base pair spacers, and again, those mark for those are our recognition signal sequence. So that tells, okay, well, this is where the splice needs to be made. So the enzyme that's responsible for the splicing is either RAG1 or RAG2. This is a recombinase, so it's an enzyme that's responsible for recombinating the genome. RAG stands for recombination activation gene, and so both RAG1 and RAG2 are needed in order for recombination to happen. So RAG1's more important because it's going to form a complex with the recognition signal sequence, and it helps to stabilize by binding RAG2. So this RAG1 RAG2 complex is responsible for the recognition. So our RAG1 is going to initiate that, and then also the cutting of the DNA so that we can splice that back together and we can exclude those gene segments that we're not going to use for that B-cell receptor. There are additional proteins, but again, RAG1 and RAG2 are the ones that are unique to lymphocytes, and there are a number of mouse models where you can have RAG1 knocked out or RAG2 knocked out, and they're not able to undergo this recombination, so you're not able to produce mature B cells or T cells. So how does this whole complex come together? So you can see in this figure how in that light gray color, that is your reg1. So reg1 is going to initiate, recognize where we have our 23 base pair spacer, and then with our reg2 complexing with it, that's going to allow for that 12 base pair spacer to be anchored also. So then we end up getting this kind of loop where we have our nomer. So that loop ends up getting spliced out. So again, we've anchored where we have our RSSs, our recombination or recognition signal sequence, and it's reg1 and reg2 that are holding that together. So what's kind of hanging off of this, off of that hairpin loop and where we have reg1 and reg2 complexing, is our J and V region that we want to keep, okay? So that when splicing happens, when we splice at where these, at the ends of these spacers, that allows for the V and the J region to then kind of bind together. In panel C, you can see some of the different components or pieces of reg1 and reg2. So again, with reg1, you can see how we have this nomer binding domain, and again, that's referring to that area where we're going to have our 23 base pair spacer. With reg2, we have this core region, which allows for DNA binding and cleaving of the DNA. Okay, so this shows you the step, what's happening to the DNA, right? So we're going to have that reg1 to complex forming, and additional proteins are also going to bind to that recognition signal sequence, the RSS. So you can see how it's binding to our spacer there. Then a hairpin loop ends up forming because we have that complex being held together. We're going to nick the DNA so that we end up getting kind of sealed. You see at the coding end how we end up sealing that. And the same thing is also happening on our J, so that when you look at step 4, again on your variable region, but also on your J segment, you're getting that little sealing that's happening. So that allows for that interwoven DNA, that hairpin loop there, to be removed. So that because it's based on those two nomers with our spacers, everything in between is discarded. So next we have to open up that DNA again. Because of the way it was cut, we're going to have hanging ends that can occur. It is possible to get blunt ends, but either way, the base pairs are such that they will end up ligating back together. So kind of think of like a restriction digest and how you cut. So that's what's happened with reg1 and reg2, is that they've cut those. These are predictable sequences, and so they are going to end up ligating back together, so that you now don't have that extra genetic information, which could include additional segments, right? So that hairpin turn of the DNA, which is going to be cut out, can include these gene segments that aren't going to be utilized. So to further add to this, these gene segments aren't always read in the same direction. So sometimes they get read in opposite direction, and this recombination can happen regardless of what direction they're going to end up lining up. So this adds again to the variability. So you might have recombination occur. You might get, a V, a J region, or gene segment selected for, but they're not going to read correctly. So there are 5 mechanisms that generate antibody diversity in naive B cell. So we were just talking about multiple gene segments, so which segments are actually put together. But there's also what combination of heavy and light chain that we end up getting. So the heavy chain for IgG with Lambda, the heavy chain for IgG versus kappa, but then within all of those combinations, we have different gene segments for kappa, different gene segments for Lambda. And so, again, you can see how that would increase the diversity. We can also get P nucleotide additions. So template nucleotides can be added at different joints. So where the antibody is going to bend. So if you think back to the classical shape of an antibody, it has that bending region. So we can change the number of nucleotides in that region. And then that will actually change and shift how the antibody is positioned. And again, that's something when I can annotate, or you can see me on video tilting my head. Just think about like the head. If you tilt your head, if you think about your head as being the variable region of an antibody and you tilt it, because we've added nucleotides, the surfaces that are going to interact are going to be different, right? At the same time, we can have exonuclease trimming, so we can lose nucleotides, so that actually changes the reading frame, so that gives us a different amino acid sequence. And then we can also have non-template and nucleotide addition. And so again, random nucleotides being added into the joints. So again, we have our gene segments. We have the different combinations between heavy and light chain. And then we can change the nucleotides, right? And so we can either do this in trimming, taking out, or adding in. And when we add in, we can add either P nucleotides or N nucleotides to cause those changes. So when we're looking at B cell receptor expression, a process called allelic exclusion occurs. And so this ensures that each B cell synthesizes only one heavy and one light chain. So instead of all this process happening at the same, there's kind of like a step-by-step process. So the first thing that happens is that the heavy chain undergoes recombination and gets expressed. Then this recombination machinery, the RAG1 and RAG2, gets shut down temporarily. And then we end up getting a light chain being expressed, but it's known as a surrogate light chain because it's not selected for, there hasn't been recombination yet, it's just expressed. And this forms a pre-B cell receptor. If that surrogate light chain is able to pair with the heavy chain, then the machinery starts back up, and so that we can undergo recombination to the light chain. So it's kind of like if they don't match, you're not going to put the effort into to moving forward because you have a bad heavy chain, right? So you put the energy in, get a good heavy chain, test it out with the surrogate light chain. If it's a good pairing, then we fine tune and fix that surrogate light chain so that we have the recombination occurring. And if that pairing isn't good, so if the heavy chain that has been selected for through recombination doesn't pair well with the surrogate light chain, then this is a non-productive arrangement, and this actually leads to apoptosis. So looking at this in a figure, we have our progenitor B cell, and again, the heavy chain goes through the selection so that we have a set VDJ for that heavy chain. If that isn't a good one, if it's non-productive, then that's where we can get cell death. Okay. If it is productive, then we move forward into producing our surrogate light chain. So then that surrogate light chain, as we can see in the green ones, they would have the pre-B cell receptor. So this is made up of both the heavy chain that has undergone recombination and also the light chain that's, just a kind of temporary thing. It's a duct tape. Let's see if it will hold. Then they're going to go through the recombination with either our lambda or kappa chain, whichever one was initiated. And then if that is a productive allele, that's a productive combination, then we have our B cell receptor, and that's what we can see in the purple cells. If it's non-productive, so if the that fine-tuning of the light chain doesn't produce a good B cell receptor, then again we would end up having apoptosis. There are chances of rearranging and trying to fix it, and so that's what you're seeing in these additional, we have non-productive allele number 1, so let's go back to the drawing board and try to fix it. Maybe we can still save it because we already got the heavy chain, right? And maybe that leads to a productive allele like we see in our purple productive allele number 2, but maybe it doesn't, it's non-productive. So again, let's go back to the drawing board, and maybe another combination would work with that heavy chain. So the heavy chain is fixed, more or less, is fixed, but we can work on that light chain. And attempts are made, but eventually if that's not productive, then that's going to lead to cell death. There's also receptor editing that can happen. So if we have a potential autoreactive receptor that when it occurs in the light chain, again, we can edit that. So again, we don't have to go back to square one. We can build upon that heavy chain that has already undergone recombination. So again, the idea is that, if the first arrangement between V3 and J3 is productive, but the combination results in a light chain where, when bound to that heavy chain, is actually would lead to autoimmunity, then they'll go back and they'll come up with a new combination, okay, before we actually, have locked in the receptor. So with B cell receptor expression, mature B cells express both IgM and IgD antibodies. And so it's through mRNA splicing that results in this, so that we have our heavy chain transcript that has the VJD put together, and then there's going to be a spacer in the RNA between it and the constant region. With mRNA splicing, we have removal of this intervening space, and that's going to end up leaving the VDJC mRNA being ready. Okay, so through mRNA splicing, this eliminates that space in between our VDJ and C region, so that it is now together. And we'll see this in our next slide. So the primary transcripts that are formed with the IgM heavy chain may result from RNA polymerase transcribing both the IgM and IgD constant regions. And so then depending upon which one ends up getting read, whether it's the IgM constant or IgD constant, that would determine which type of antibody that you end up producing. So again, as you can see here, we have our VDJ that has been selected for. So we've removed all of that, those nucleotides in between, thanks to RAG1 and RAG2. And then we have these two constant regions that are present. One would code for IgM, and the other one is going to code for IgD. Okay, and so then depending upon which piece gets read and transcribed, that's how we end up with these two different mRNA fragments. So with mu, which is the one on the left, it has a like U Greek letter, that's going to make the IgM. And then for delta, which is on the right, that would make our IgD. And we can have it where they're secreted, or we can have membrane bound. So we can see in the membrane bound, there's going to be a portion of it that creates these alpha helixes that are transmembrane. That portion has been excluded, has been cut out in order to have our secreted antibody. And so again, it's through this mRNA splicing that allows that to occur. Okay, so we're going to switch gears and look at the T cell receptor and its genes and expression. So just some assumptions that we can make when looking at T cell receptor genes. T cell receptors are only going to be expressed in T cells. T cell receptors are membrane-bound proteins, so we don't see them secreted like we do with the B cell receptor. T cell receptors genes are also going to undergo the rearrangement like B cells to lead to increased diversity. And like immunoglobulin genes, like B cell receptor genes, we're going to see where we have constant and variable regions. So we're going to have regions that stay the same, and then we're going to have regions that are going to undergo the gene segment rearrangement so that we can get the different variabilities occurring. So just looking back at a figure from chapter 3, we looked at the T cell receptor and we talked about how there's two chains or two subunits, and within these subunits we had the constant and variable region. In panel B, looking at the ribbon model, you can see how these domains are formed, how we have the domain making up the constant, and then we have our domain that's making up our variable region. If we look closer at that variable region, we'll notice that there's these loops that act as receptors, CDRs. So these can be receptors to different CDs, but also this is where these peptides are going to end up binding to, and that these are the portions that we're going to kind of change up so that we can bind to unique peptides also in this variable region. So looking at the T cell receptor, again, it's going to be made up of two different chains. So we have alpha, beta, gamma, and delta. And so on the top one, starting at the top in panel A, we're looking at the alpha and delta, and then moving down the beta, and then the gamma. Okay, and so you can see how those components that we saw in the B cell receptor, where we have these variable gene segments, in some cases we have D segments, we have J segments, and we also have the constants. Okay, and you'll notice that for some of these, those J and C are paired, but then we're going to be selecting and getting our different receptors. So again, the process is similar to what we saw for the B cell receptor, that there are going to be these recognition signal sequences, RSSs, that are going to be located that allow for identification of where the gene segment is, so that the gene segment isn't accidentally kind of nicked out, so that we can get recombination with these different segments. It is possible to get more than one segment, so you can have two D segments. That's possible, and that's been observed for the delta gene, but for beta, it's possible, but it has not been observed. So looking at allelic exclusion, T cells are able to undergo this, but it's not the same machinery that's utilized as with a B cell. The process of T cell receptor gene expression is tightly regulated. So we have these enhancers and promoters similar to what we see in the B cell. They're also found in T cell alleles. We also control gene expression during development, very similar to with B cells, where we see like a pre-T cell receptor, and then whether it gets produced, production in the screening looks for functional rearrangements, and that determines whether the cell will end up surviving or not. So this table's from the textbook, and it does a nice job of comparing the processes between the T cells and B cells. A little overwhelming, but if you take each line and kind of work through and think about, you know, the information presented in the audio lecture or from the textbook or from just the slides, we can start to compare and contrast these processes. How are they similar in B cells and T cells, and how are they different? So when we look at these gene segments, they're present in both T cells and B cells. We have light chain segments in B cells, which would be our kappa and lambda, that only have VJ. Similarly, we have chains that only have VJ in T cells, and that would be alpha and gamma. When we look at the heavy chains, for B cells, we have a single heavy chain with V, D, and J segments. With T cells, we have beta and delta that have that V, D, J segments. For both B cells and T cells, we need to have Reg1 and Reg2 expression. And again, this is where if we have a knockout mouse, we can see how they cannot produce a B cell receptor or a T cell receptor. Also looking at increasing the diversity, we can have the addition of P and N nucleotides in both T cells and B cells. Looking at whether there's multiple D regions that end up in the recombinated chain, what ends up becoming the receptor. That does not occur in B cells, but it is present in T cells. Again, only observed in the delta chain. Keeping in mind that the only chains that have D regions for T cells are delta and beta. And again, in beta, we don't see this. This hasn't been observed, though, you know, theoretically it is possible. Allelic exclusion happens in both T cells and B cells. In B cells, it's an absolute happening, but because of the difference in the machinery, it's not always absolute. Especially when you look at that alpha gene. When upon activation, whether the receptor gets secreted only occurs in the B cell. This does not occur in the T cell. The T cell receptor is always membrane bound. And then looking at the constant region, for B cells, that constant region determines the function, what happens, because the antibody gets secreted, other cells can bind to that constant region, and depending upon what constant region, it can bind to different receptors that give you different signal transduction pathways activation. So you would end up getting different responses by other immune cells. That constant region in a T cell doesn't interact with other cells because it's membrane bound, so it's only going to it's anchored there, so it's only going to initiate signal transduction in the T cell, and that's pretty fixed. Okay, so then looking at a process called somatic hypermutation, which we didn't talk about because we haven't activated our cells yet. We just got the initial, how do the receptors get expressed? But after exposure to antigen, B cells can undergo a process called somatic hypermutation, which means they can further edit and change the receptor. And this allows for the receptor to have higher affinity to antigen. So if the B cell sees antigen, it fine-tunes its receptor, its antibody, so that it can bind stronger to the antigen. T cells don't do this. Once they've made the receptor, the affinity is what it is, okay? So again, this table does a nice job of kind of summarizing up a lot of the information that's in this chapter, and then look for the second audio lecture that is going to be on MHCs, the other receptor, major receptor in the adaptive immunity.